Good afternoon or good morning to everyone, wherever you might be. We'd like to thank you for joining this first of a series of webinars that we would like to organize here at Biotrial on the subject of EEG in drug development. We're right on time right now. It's 4 p.m., but we'll probably wait another minute or two for any latecomers to join us. In the meantime, just a few words on Biotrial for those of you who don't know us. We are a full-service CRO with close to 30 years of experience in drug development. We provide services to biotech and pharma clients for many aspects of their development needs. This includes preclinical in vivo work, for example, phase 1 clinical research, phase 2 to 3 trial management, and we also offer ancillary services such as data management and biostatistics. Today's presentation uses much of the experience that we've gathered in our core lab unit, which manages the central reading of various types of exams, our particular interest today being EEG, of course. The presenter you'll be listening to in a few moments is Dr. Philippe Danjou, who's an MD and PhD. Philippe has over 30 years of experience in academic and industry research. He has held several positions within clinical pharmacology at various groups such as Synthelabo, Wyeth, and Merck. Today, Philippe is currently our Senior Director of Research and Development, and as such, he acts as a scientific reference for our clients' CNS projects. He also leads our internal R&D efforts to develop novel CNS research models, something that we're very busy and active at, CNS being really one of our main areas of expertise here at Biotrial. So we can start in a minute. Just couple things we wanted to let you know. You feel free to ask any questions using the webinar tools that we have here today. We'll have time for a Q&A session at the end of the presentation. We'll do our best to answer all the questions that we receive. Any questions that we don't have time to treat, we'll be glad to follow up with you on at a later time. Having said that, I will hand it over to you, Philippe, and wish you a nice presentation to everybody.
Thank you, Mathieu. So my duty today will be to show you the benefit of EEG in the frame of drug development and how it links to the technical aspect of progressing a drug through its life cycle. So, as we all know, the success rate from the beginning of phase 1 to registration is pretty low, and that is the latest survey we could find by Thomas and co-worker, which studied about 10,000 transition of phase over 7,000 drugs. So as you can see, all indication pooled, there is a likelihood of approval of about 10% from the beginning of phase 1, which goes down to 8% in neurology and 6% in psychiatry. So the domain of CNS is, as we all know, a domain which is at risk of failure in drug development, reason for which the best countermeasure should be taken. In the same survey, there was a breakdown per type of studies which have used biomarkers and those who have not used biomarkers, mostly at screening. So that does not necessarily mean that this biomarker would have resolved the issue, but at least understanding the underlying heterogeneity in biology or in the physiology of the disease is important. And when a biomarker has been used, the success rate has been tripled from the drugs without a biomarker. So that seems to be an important documented factor. What I will be doing today is actually addressing the development with 3 big boxes, one being preclinical models with animal studies, one being early trial, including phase 1 and and proof of concept. And the third one is actually the real life with phase 2b and phase 3. Also, the costs are increasing from the beginning to the end of the development, reason for which it would really be good to have a biomarker or a tool to help in progressing the drugs in order to de-risk a little bit the investment for the next phase. So that could be an early kill, or that could be a better drug definition for the dose. And eventually it may a bit de-risk the whole process, which otherwise is pretty discontinuous. So what do we mean when we are talking of about biomarkers? We can mean very many things. And really it can range from a read very close to the receptor, like, for example, the displacement of a PET ligand, which has no direct relationship with what occurs downstream. And it can also be very close to the therapeutic response, almost a surrogate of efficacy. The EEG and the ERP stand somewhere in the middle of that spectrum, not at the receptor level, not at the level of the response, but in between. Also, in the drug development, as we all know, there are some differences between species. For example, rodents have a smaller frontal lobe than humans, but on the other hand, humans can speak. So all of that could be taken in order to maximize the benefit of drug development and to validate biomarkers. One important aspect of the early trial in the central box I've just shown is the fact that in healthy subjects at variance from patients, there is sometimes no dynamic range for an increase or for a decrease of something. And that dynamic range can be created by disease models, which is a way to create a space for improvement for a drug, and we'll see a few examples of that. Also, one important component is the fact that biomarkers are not just something that you can piggyback on an existing study or that you can plug as an add-on, but it may modify profoundly the design, including the sample size. So the sample size is really key to the successful use of a biomarker, which cannot be taken separately from the rest of the concept. So, in one word, to understand completely the biomarker, it should be viewed globally throughout the development, but not from a silo. So we'll consider the relationship between these boxes and where the biomarkers can help. When you are at the early stage of development, when models of pharmacological efficacy or read of the physiology is being used, at this point, you would really like to know if the next trials to come, which is a phase 1, is going to show if the target is engaged and at which dose. So that, for example, can be in the scope of EEG when in animals PCP is used to produce a response. It does produce a very significant response on EEG. And the equivalent in humans would be ketamine and another NMDA antagonist, which would produce an increase in the resting gamma, in induced gamma, and in ASSR, standing for auditory steady state response. On the other hand, in animal models for schizophrenia, very often people use chronic PCP, which produces impairment and alteration of the parvalbumin-containing neurons by producing an oxidative stress on them. And that is really not that is really not applicable to humans because that is de facto a lesional model in a bit similar to MPTP. Conversely, from the clinical pharmacology studies, which are now relabeled translational medicine, there are some important findings which can be applied to animals. So that is a bit a conventional way to modelize a disease and create an animal model, like we do for stroke, hypertension, inflammation, etc. But there could be some specificities which are used in clinical pharmacology, which can trigger some new models in animals. So to give an example of that, we've been using a human panic CCK model, which has led to the development of an alias in animals, which is a CCK potentiated elevated plus maze. So who would have thought of developing such a model if there hadn't been a human models? Similarly, for the human CO2 anxiety model, which also has turned into a rat model, and CANTAB, which was developed to be used in animals and in humans, etc. So one more flux between these boxes is going from a successful early trial to a phase 2B or 3, and that is really depending on the predictive value of early trials. It is a relatively complex area where the validity is high for Me Too drugs because there is a lot of examples and you know which kind of expectation you can have based on an early read. And it's also more valid when it's mechanistically mediated rather than symptomatically driven. We all know examples of parameters that change and which are predictive of the efficacy. Receptor occupancy is a good example for Me Too when you know which kind of occupancy you need to reach. Effect on sleep also propagate very well from the models of insomnia to insomnia, but also analytes like cholesterol, blood pressure, or in the case of EEG, the relative magnitude of an effect on a predefined EEG band for a Me Too. So that for a benzodiazepine or a SSRI, for example, you would be able to tell in which range of those is the probable efficacy based on early trials. Last is the final arrow, which is creating human models from the pathology. So it is very similar to creating animal models, but it allows more precision with larger images, more electrodes, and also a subjective information on what the subject feels. So there have been numerous examples of that in the field of psychiatry, for example. Chemical depletion in monoamines, virtual lesions with transcranial magnetic stimulation, theta burst, now anodal currents, functional testing with imaging with a given paradigm, cognitive testing, disease models, pain models. Pain models, for example, are trying to mimic the neuropathic or inflammatory or excess in nociception component of pain. So where are we with EEG in that area? So that is a technology which is low-tech and inexpensive, which can be used in human, rodents, dogs, and non-human primates. Some of these species have particularities. So for example, dogs allow, because of their size, to implant a catheter in cisterna magna and to obtain CSF at given time points. There is in general no underlying hypothesis for resting EEG. That is not the case for event-related potentials. And importantly, it creates a multi-array by measuring during numerous physiological conditions the signal we're interested in. So for example, you would measure EEG in eyes closed and eyes open condition, but you may also do it during the night. And during the night, the different sleep stages and sleep cycle would allow to have read on different processes active at different time of the circadian cycle. And it's even more complete if ERP are generated based on the requirement. So there are two types of signals which can be collected. Some are oscillations, which are collected by the electrode in an unconstrained resting state, and that is just observational. Or there is a possibility to produce potentials triggered by a task, which is averaged in a time-locked manner to the stimulus. And that is reproduced hundreds of times, and the response is average, which explains that the grand average is stable. The potentials which occurs or would be picked up as a random and not being time-locked are not, we don't know how to analyze them. They may represent some of the neural code, but there is no way to know what it does. So basically, it's either oscillation or potentials which are time-locked to the stimulus. So when you look at how the EEG works and how it collects potential, the electrodes, as you can see on the left part, are affixed on the skin. And to connect electrically with the cortex, the signal has to go through the skin, through the skull, and through the membrane surrounding the brain. So basically, it is not an acute information on the cells which are just below, but it's more a vectorial integration of a region in the vicinity of the electrodes. And to make it simple, we can say that it is within 1 centimeter range around the position of the electrodes. So the more electrodes, the more spatial resolution, but the spatial resolution is not always needed. Where do these potential come from? They come from the synchronous depolarization of a number of neurons. And for the oscillations, these also propagate between short distance or long distance communication between cortical areas with different frequencies. And one of the important sources of oscillation are the so-called corticothalamic oscillators, which are loops which are formed between the cortex and the thalamus. The neuronal composition of these oscillating circuits is known since the 90s. And there are 4 neurons involved in that reverberating and oscillating circuits: the pyramidal cells, some GABAergic interneurons, and 2 types of cells in the thalamus. So this is what is called the thalamocortical loops, and it is a widespread generating system. Now, there are local oscillatory circuits in the cortex, which are based on the pyramidal and the parvalbumin-containing cells, which inhibit the pyramidal cell at a fast pace, producing some resonance between the circuitry and the frequency, which peaks around 40 Hertz for humans. And that is what is called the microcircuits, and which produce that vast peak of amplitude at 40 Hertz used by ASSR. For the oscillatory components of the EEG, this is a survey of a sensitivity matrix in the rat, and what is presented at the top are the frequency band, delta, theta, alpha, beta slow, beta fast, and gamma. As you may see, the gamma is not always represented because gamma has been lately of interest, but 20 years ago, no one was interested in gamma. So that is a recent, between bracket, a recent acquisition. And what is presented here is the increase, decrease of the magnitude produced by a large number of drugs in the rodents when they are awake. So, for example, we'll take the allosteric modulators of the GABA, like the benzodiazepine, which produce slow delta and theta wave that decrease alpha and which increase the beta band. In humans, it's the same sensitivity matrix. And when one looks at the GABAergic modulator like the benzodiazepine, you have a similar pattern with an increase in the delta, a decrease in alpha, and an increase in the beta band. Gamma was not studied at this time. So here is some work which was done at Syntelabo with diazepam, which is presented on the x-axis of the left panel is the frequency in hertz. And at baseline, you see the alpha of the rat, which is in the frequency range 4 to 6, which when lorazepam is injected is suppressed while fast activities occur, and a little bit of slow activities. And when the antagonist annexate is injected, there is a reversal of it with a comeback to the initial resting state or the initial spontaneous electrocorticogram recording. When the same experiment is done with butobarbital, you have a change in the spectrum with fast activities, but it's not reversed by the benzodiazepine antagonist as expected. And when one looks at zolpidem, which is a selective modulator of the alpha-1 subunit, then you see a slightly different pattern with some suppression of the alpha, some slow waves, lots of slow wave, and a little bit of fast activity, slightly different from lorazepam. When we did the studies of zolpidem at 5 and 10 milligram and 20 milligram in the healthy subject, and again, that axis represents the frequency band, and the time is here over 8 hours, and it's the data are expressed as change from baseline. So what you can see in the placebo condition is a relatively stable energy in the different bands over time, whereas with zolpidem 5 and 20 milligram, you see a very similar pattern as the one I've just shown in the rat with increase of slow activity, suppression of the alpha, and some fast activity, which are a little bit proportional to the dose, but not so much. The way it occurs when you are using EEG in the first in man, and that is posing the problem of the sample size and the effect size, which in general you're not sure about the effect size you're going to collect, but this is an example of a first in man study with parallel groups in which EEG has been used. And in the third dosing group compared to placebo, you see some hints of changes as expressed in false color on a flat mapping manner, which in terms of p-values, these are the same maps, but the color coding is for the p-value. You can see that there is actually a change which occurs in the gamma at the top dose. And that is really of interest because the signature of the drug in animals was an increase in the gamma in a bell-shaped manner. So that study is ongoing, and we'll see in the future if, as in animal, it then goes down, or if, as very often when you go to animal to humans, a bell-shaped curve does not turn into a bell-shaped curve. The same type of information is available for sleep, in rat and in humans. I will go rapidly on this, just an example of a similar sensitivity matrix. And sleep has a property interesting in the domain of psychiatry, and that is that when one looks at the effect of existing antidepressant, or the latest one, I'm not totally sure about it because that survey was done before, but all of the other antidepressant, when you look at the REM suppression produced by a single dose or a multiple dose of an antidepressant, its predictive value for a successful registration is when a single dose is positive 76% out of 21 studies and 84% out of 13 multiple dose studies. So that is actually in excess of a positive phase 2 as a predictor to registration, which has some false positive and which has a predictive value of 60 to 70%. So even if it is not what you are primarily interested in depression, the REM suppression is an important biomarker of the efficacy of antidepressants. So we have covered the spontaneous oscillation of these cells depolarizing together. And we're now rapidly going to discuss the evoked related potentials, which are a very vast domain. And there is some degree of consensus on the most frequent one, but it's still debated for the exotic ones. And again, the standardization is important. So I will just mention some of them. P50 is what is called auditory gating. It is when you are listening to two beeps in a sequential manner. When the second one is warned by a previous one or not, the response is decreased when one is warned. And that is something the normal brain does very well. And it's something that the brain of patients with schizophrenia does not do at all. So there is no warning effect of a previous stimulus. Mismatch negativity is a parameter which, by which the brain reacts to the violation of a rule in an unconscious manner. So for example, when you need a series of beep and there is an infrequent one which is slightly different, when that occurs, a specific negative potential occurs without the need of paying any attention to it. Again, that is altered in schizophrenia, Alzheimer's disease, bipolar disorder, etc. P300 is using a visual or an auditory stimulus with an oddball paradigm, which is very similar to MMN in the sense that where it used a frequent stimulus and a deviant, but here the attention has to be consciously attracted to the paradigm, and it is a top-down process where attention modulates the response of P300. So it is close to it is a late cognitive potential, and it's altered in several disorders, including Alzheimer's disease. I will not discuss very much the valence-associated ERPs, which are under research and which are believed to read something involving reward. And I will discuss a little bit more the auditory steady-state response, which is the response of the brain to a train of clicks at the frequency around 40 Hertz, which evoke the resonating property of the microcircuits composed by a pyramidal cell and a parvalbumin-containing GABA interneuron. So this is a study which we conducted for a client in which trains of clicks at 40 Hertz were applied for 400 milliseconds, lots of clicks, and the auditory evoked gamma, the power in the gamma band, was measured during that phase. So this is at baseline, where the placebo and the active drug, which was escitalopram 15 milligram, were overimposable with a decay over time, like in animal species. And when the drug was introduced, there was on that evoked gamma a high magnitude potentiation with a much larger response, the standard error of mean being materialized by this red shaded area. And it did continue on day 3. It was so this 3-day sequence meant to produce therapeutic plasma levels. So there was a roughly similar response which was obtained on day 1 and day 3 with no obvious tachyphylaxis. So what are the practical translational aspect of all these biomarkers between rat, monkey, and man? The P300, of course, is obtained in man and may be obtained in the monkey, but it has very rarely be done, needing a training, and it's not possible in the rat at all. For mismatch negativity, there is an equivalent in the rodent of some sensory discrimination with an echoic memory component, which relates to mismatch negativity. It is not mismatch negativity, it's not exactly the same process, but it has some mirror image. The same MMN exists in monkey and of course in man. QEEG, there are a few between species differences. I will just give an example. The biology is different for the 5-HT1A receptor and produce different effects between rats and mice, for example, the autoreceptor producing opposite direction effects. Monkeys and the other animal species are useful, and some big companies sometimes do 3 species like rat, dog, and monkeys to incrementally increase the precision and get closer to the human species. So that is not necessary, but between nothing in animal and all the species, there is a range. And ASSR, auditory evoked gamma, is produced in all species, including rats. So EEG is a biomarker that can follow across drug development at early phase. In preclinical, in early phase, a special care has to be given to the power calculation. And even in the late phase, in a multi-center setting, especially when the clinical evaluation has been deceiving in the past, like in the Down syndrome or in Alzheimer's disease, where a convincing add-on may be really useful for decision-making purposes. It can also help in phenotyping subpopulation in a personalized approach medicine. And we believe that with genomics will be increasingly important as the survey about biomarkers proved to triple the success rate. It has a low cost portability, and it's very precise related to time if one wants to do PK/PD. So as a conclusion, I would say that there are 2 frequent scenarios. One is when a signature is found throughout in the preclinical research, and it is also existing in humans, then it paves the way for a PK/PD modeling for the dose and for the time course, and can be very useful for determining the human target engagement. On the other extreme of the spectrum, when an ERP of interest exists for a given pathology, then efforts should be made to apply it and de-risk a little bit the drug development based on that ERP, which presumably would be closer to the disease mechanism like P300 for cognitive impairment. And that is these are the quick facts I wanted to provide you with. and I think Mathieu will now take back the lead and address the question that you may ask with the tool of the webinar. So I thank you for your attention. And if you need access to these slides, we would be more than happy to provide you with. Thank you.
Thank you. Thank you very much, Philippe. So if you can just go to the next slide in the presentation. One thing that we did want to ask all the participants today for feedback on is potentially the next topics that we would discuss during future webinar sessions. So you can see the topics here that we are thinking about. The first one, EEG, clinical and non-clinical operations. This would be looking at the logistics and, let's say, the challenges of implementing EEG within preclinical and clinical trials, phase 1 and phase 2, phase 3. The second one, EEG for safety. So how to use EEG as a safety marker, looking at proconvulsant effects, for example, or epileptogenic patterns. EEG pre-processing and artifact rejection would be something on how to treat the signals to be able to gather the maximum information possible from everything that we gather during the EEG recordings. The QEEG spectral analysis, this I think speaks a bit for itself. Sleep EEG or polysomnography is also something that we might consider discussing during the next session, and ERPs, since there is a wide variety of ERPs out there. We think that this could be a topic of interest. So we've shared so we're sharing the question here using the webinar, and if you want to give us an idea of what your thoughts are on this topic, we'd be very glad to have that information. Philippe, I think that there are a few questions that we've received from the participants. Could you take a few minutes to address those at this time?
Okay. So the first question is, can you do any simultaneous recording of EEG and functional MRI? And the answer is yes. It requires some relatively specific equipment, which would not create currents in the field of the gradients, but it is absolutely possible, and it's even complementary, because the functional MRI would give you a good spatial read, but not a temporal read, while the EEG would give you the precision over time. The second question is, for ERP, what is the best option for stimulation, visual or auditory? The visual and the auditory system are really organized in a very different manner. So very often the auditory system is more used because it is automatically treated. It doesn't require that much of attention, and it's not organized in increasing the contrast, which basically in the visual system, it's organized to make the unusual stimuli disappear and the usual one to be contrasted up. It's also more complex to be used in rodents. So in general, auditory is much more frequently used for all of these reasons. The next question is it possible to assess deep brain sources with EEG? There are some methods which analyze where the sources are, the reverse problem, as it is called. There is a level of imprecision with that. So anything in the depths of the brain has some imprecision in the way to localize the source of the oscillations, whichever method is used, and the most common is Loretta. Next question is, can you do a QEEG analysis on an EEG capture for safety monitoring? Yes. When safety monitoring is done, it is in general a discrete capture of sequences of 10 to 15 minutes, and you can do a spectral analysis. You can do whatever you want on it. The only issue, which is true for both safety EEG and spectral, either FFT or wavelet or whatever, is the sample size, because very often these studies are done in parallel groups with a limited sample size, and it is not necessarily adequate for a low effect size. On the other hand, some people have done crossover study or incomplete block design in the phase 1. For example, some of the big American companies do not do parallel group study, but do incomplete block design in order to control the within subject variability. And by a minimal increase of the sample size, the power is really boosted. So it's all dealing with the question of power calculation, that answer. Is there any interest for deep learning approaches in EEG data? Yes, absolutely. That is really going to be the future, and people have already started to do it with limited in limited domains of knowledge. But it's pretty sure that in the future there will be new data or data will be analyzed by deep learning and will eventually succeed where people have failed 30 years ago and have overpromised with the pharmaco-EEG classification. Do you have examples of use of EEG in pharmacological models? Though I suppose that means in animal models that would be conducted with a specific paradigm. Are there some recording that can be conducted and from which you could synchronize with a task? Yes, that has been done. For example, as I mentioned, the equivalent of MMN is existing in rodents, but it could be generalized. It's mostly a matter of standardization and of limitation of the animal brain for what you want to measure. How reasonable is it to use EEG as a predictive marker of convulsions? Again, it is well, it's a complex question, because by definition the convulsion when they occur are different of the state in which the brain is not convulsive. So there could be there are some indicators that a seizure is close to occur, or that there is something abnormal pointing in the direction of an hypersynchronous discharge. However, it documents the fact that there has not been epileptogenic associated traces. Would it allow to detect a seizure before it has occurred is a bit of an unresolved question. And the general belief is that it adds some value, at least to document the lack of. On the other hand, if I want to be completely honest, I've seen cases where the EEG was recorded, and 1 minute after the subject has a tonico-clonic seizure, and before that the EEG was normal. So there are arguments pros and there are arguments cons. Basically, it is a regulator which requires it in general to show that there has not been something overlooked. What exactly in the MMN in terms of standard, difference, and deviant waves is the difference between rodents and humans, and to what degree does it translate? We're a little bit at the beginning of that. We are pretty well in control of the MMN in humans and the effect of the properties of the deviants. For the rodents, it is less clear, and I would defer a little bit that to the another seminar which will be focused on the ERPs and which will show the different paradigm used in animals in order to create some echoic memory similar to mismatch negativity. So I will defer the answer to that to the next seminar. I apologize. Mathieu, have I addressed all of the question, or should I still try to go down? Is there something
No, I
more
think
down? No?
No, thank you, Philippe, and thank you, Philippe. I think we're all set. So I think we just need to thank everybody for joining us today during this webinar. If any further questions do come up, please do feel free to send them our way. My name was Mathieu Grace, by the way. So you should have my my email address in the invitations. Please do feel free to reach out if there's any other topic that you wanted to discuss. With that, I think we will end this this webinar. There's a couple questions that should pop up when you do close the webinar. Please do feel free to answer them, as it'll help us to make sure that the future sessions are of the utmost interest to all participants. Once again, thank you all for attending, and we wish you a very nice day. Bye-bye.